Hello everyone, how are you? Today we are going to pass a new topic, 13.5 chapter, Genetic Engineering, page 182. So, genetic engineering, how to insert genes into an organism and produce new organisms. Today we will pass about this kind of processes. In nowadays, there are two main development of technology. It is one, biotechnology, and second one is nanotechnology. So in biotechnology is related to the biology, that's why we are passing about it. And by the help of this biotechnology, we can do progresses in the technology by using some organisms, some genes, DNA, chromosomes, and make some changes to get a new clones or to get a new type of organism, and we want to develop this technology. So what is genetic engineering? It is a set of research methods that make possible examination of particular genes and introduction of changes into them in order to modify genes, and it is necessary to isolate DNA from the biological material, fragment it, connect it with the carrier, and then introduce it into the cell of recipient, in whom it will be expressed together with his or her genetic material. So genetic engineering involves transfer of the gene from one organism to another, and it should be expressed, produce the proteins in a new host organism, guys. There are many branches in genetic engineering. It is used in many ways in our life. For example, in medicine, in agriculture, we use some plants and animals organism to produce genetically modified objects. In environmental cleanup, for example, in biofuels, we use the genetic engineering, and we use it in industry production, for example, to produce washing powders, dietary products, and etc. The plasmid, it is the circular DNA particle found in bacterial cells, and it is transmitted to the gene of immunity to antibiotics. And there are a vector. In genetic engineering, it is a DNA particle which enters the cell and replicate in it. Then you should know the terminology like restrictase. It is a restriction enzyme which helps to cut fragments of DNA. And the last terminology which you should know, it is DNA ligase, which helps to stick the ends of the DNA, and it is a short length of unpaired bases. Let's watch this video about restriction enzymes and DNA ligase.
In order to recombine DNA, it is necessary to divide it into smaller fragments with the use of restriction enzymes. These enzymes identify particular sequences within the DNA, and then cut DNA within those sequence boundaries. The sequences are often palindromic, which means that the order of nucleotides of one strand is identical with that of the second one, if read in the opposite direction. Certain restriction enzymes cut both strands of the identified sequence of DNA in places located precisely opposite to each other. In such a case, the so-called blunt ends are created. However, the majority of restriction enzymes cut both strands of the identified sequence in locations shifted in respect to one another. The formed fragments have single-stranded, so-called sticky ends.
If the DNA derived from two different organisms is cut with the use of the same restriction enzyme producing sticky ends, then fragments with complementary ends are produced. By mixing these fragments with each other and adding ligase, the recombined DNA is obtained.
The vectors used most often are plasmids of Escherichia coli bacterium, containing 3 necessary areas: the site of replication start, the gene of immunity to antibiotics, and a site onto which the DNA fragment can be embedded, without distorting the replication of the plasmid and gene expression of the gene of immunity to antibiotics. Plasmids can also be vectors introducing alien DNA to eukaryotic cells such as YAC in yeast, TI in plants, or SV40 in animals.
Жалпы қысқаша айтып өтсек, гендік инженерлік технологияны қолдану адамның өмірін жақсы дамытады және оны оңайлатады. Мысалға айталық, қанттық диабетпен ауыратын адамның инсулинін өсімдіктің ішіне салып, сол жерде өсіру. Сөйтіп одан бір дәрі-дәрмек жасайды да, осы технология арқылы. Мысалға, Poplar Trees деген фактіні көре аласыздар 182-ші бетте. Сол жерде айтады, ол ағаштарды әдейі судың жағалауына егіп қояды, өйткені өсімдіктің тамырлары судың ішіндегі зиянкесті молекулалардан тазартып отырады екен. Және ол судың ертеңгі күні буланып, ауаға жаңағы токсикалық заттардың шығып кетуіне жол бермейді екен. Тағы да бір пайдалы жақтары ол біздің агрономияда. Мысалға, өсімдіктер өсіруде немесе асыл тұқымды жануарларды мысалға селекция жасауға, өндіріс жағынан, әртүрлі порошоктар жасауда, дұрыс тамақтану жолында қолданылуға келетін өте көп пайдалы әсерлерін тигізеді. Бұл генетикалық инженерлік технология. Сондықтан біздерге бұл өте маңызды. Thank you for your attention. See you soon.